in order to search the mass spectral data against. And to do that, we took a commonly used food frequency questionnaire, and we took all the dietary items and we divided them into their genera. And for each genera, we took protein markers that are specific to that genera. We weren't interested in very fine phylogenetic resolution in diet because we didn't care which type of cucumber or fish, exactly type of salmon that the host consumed, just to know that it ate salmon or cucumber. And here in the middle, in the insert, you can see the number of protein markers that were available for each dietary item. And here you can see them more outlined in a column figure. So there are a number of protein markers that represent each food item. And these are the absolute numbers of different type of proteins. In general, across the presentation, throughout the presentation, the host proteins are represented in blue, the bacterial proteins are represented in red, and the yellow are representing dietary proteins. So then we just did a few sanity checks just to see that we are measuring what we think we are measuring. We look at two different human cohorts of Israeli and German individuals. And we started looking at different food types just to see that what we can see, if there's like a broad common signal that we can characterize. And we saw that in terms of bread consumption, looking at the protein derived from meat, there was no significant difference in the stools of those individuals, of the two different populations. But when you look at pork, which of course is not being routinely consumed by most Israelis owing to cultural and religious reasons, we saw much more abundance of pork proteins in the German population, and the signal was derived from several different pork proteins that you can see here. Could you, by the way, see my cruiser on the screen?
Yes, absolutely.
Thank you. As for chicken, there was slightly more consumption, or maybe consumption is not the right word because these are just remnants that are found in the stool, but we saw traces of higher abundance of chicken in the Israeli population. Chicken is being consumed very commonly. We did a dose escalation study for individuals who consumed, just for healthy individuals who consumed varying amounts of peanuts. And we saw that our pipeline was able to detect this dose response curve. When people ate more peanuts, we saw more traces of peanuts in the stool. And again, there's a complex interaction here between consumption, absorption, and the traces that we see, of course, in fecal samples. So talking about absorption is a bit problematic, but we will do an attempt later to address that issue. We did another dietary interventional study in which we let individuals consume two different food items as a pair in a pre-specified period. And we used two different pairs of food, bananas and apples, and then peanuts and cucumbers. And we did see that during the intervention, we were able to detect those food items in the stool using our pipeline. So now we have a pipeline that kind of simultaneously analyzes all the metaproteome from the GI tract. We can see host proteins, bacterial proteins, and dietary proteins as well. We next moved to analyze the signature of IBD. So we took cohorts of IBD patients. This is a cohort of Israeli pediatric Crohn's disease patients. And we were able to, using the pipeline, we were able to identify different host proteomics and microbiome proteomic signature that was significantly different between healthy and controls. The same was true for a German cohort of ulcerative colitis patients. For both Crohn's disease and ulcerative colitis patients, this of course has been known from before. There are multiple antimicrobial peptides that are upregulated in subjects with IBD. We also identified a few candidate antimicrobial peptides, and here we only labeled just a few of them, but there are much more that are hiding and not labeled here. When we did pathway enrichment analysis for the microbial proteomics, we could see that both ulcerative colitis and Crohn's disease had a dysfunctional or functional dysbiosis at protein level. And we could see a few pathways that were enriched in subjects that were with the disease compared to control. One interesting finding that we saw, and we were not aware of, and as far as I'm aware of, this was never demonstrated before, alongside with the upregulation of post-antimicrobial peptides in both ulcerative colitis and Crohn's disease that I've shown in a few slides back, we saw an interesting observation in which we saw that the vast majority of commensal protein were underregulated. So here, for example, these are just two exemplary bacteria. I'm going to show you the global picture in a second. But just looking at those two volcano plots, you would expect to see a balanced volcano plot in which the right side and the left side, which are proteins that are, each dot here is a bacterial protein, you expect to see upregulation or downregulation of bacterial proteins that go either way, either upregulation or downregulation, but you see that the vast majority of proteins are downregulated. And this was true across disease, across cohort, across geography, and across species. Here you can see that all the differentially abundant proteins, and you can see that most of them belong to proteins that are downregulated in patients or upregulated in controls, relatively. You can't really determine, but let's say relatively downregulated in IBD patients. Those that are marked with the arrows and the plus are species that were not identified by metagenomics when we did differential abundance analysis on, conventional metagenomic data, suggesting that using metaproteomics to identify disease-associated bacteria may reveal disease-associated candidates, bacterial candidates that you wouldn't otherwise find if you just use genomic readouts. This is all the species combined, grouped by the disease, and you can see that there are far more differentially abundant proteins that are upregulated in healthy controls or downregulated in IBD patients than you would expect in like a balanced scenario. So basically, there's like in general global bacterial protein synthesis inhibition in IBD that was not previously known. Next, we said maybe diagnosis and monitoring of IBD is something that can be quite tricky in clinical practice, and we said maybe we can identify novel biomarkers to identify disease activity or diagnosis. For that, we used publicly available samples from the human microbiome cohort, and we tried to identify a signature that's associated with IBD at protein level that would signify it is the diagnosis. And sorry for the small labels, you can see here that we identified a lot of potential biomarkers for both Crohn's disease and ulcerative colitis that performed a lot better than the most widely used protein in current clinical practice, which is calprotectin, which is composed of two subunits called S100A8 and S100A9. And we did it across validation cohort. We had a training cohort and a validation cohort, and we used proteins from both host and bacteria, and we were able to identify protein pairs that outperformed calprotectin, again, which is the gold standard today in clinical practice in the diagnosis of IBD patients. We were also able to determine compliance of patients with diet and the clinical response to diet. We took patients who had recent diagnosis of new onset Crohn's disease, and for pediatric patients, one of the first-line treatments is exclusive enteral nutrition, and those patients were enrolled in a trial in which they commenced exclusive enteral nutrition, and we sampled them before and after commencing on the diet. And we were able to see a reduction in the number of dietary proteins that we could detect, as expected, and a correlation between basically the diet and the inflammatory signal, suggesting that after treatment, the inflammatory signal in the gut, here signified by, again, one of the subunits of calprotectin, is reduced. Here is a cohort of celiac patients who commenced on again, new onset celiac patients who commenced on a gluten-free diet, and we looked on gluten-derived proteins, gluten-related proteins. And we were able to identify one patient that wasn't compliant. We also had, we known it from the Food for Food 3 questionnaires that they filled, they was not compliant with the diet, suggesting that this pipeline can also be used as a measure of assessing compliance with the dietary recommendation in clinical practice or medical practice. And last, as I said, I want to just touch the absorptive function of the gut, and whether we can say something on that using the pipeline. Again, we took two cohorts from the Human Microbiome Project, one from the Cincinnati Hospital and one from the Massachusetts General Hospital. These are two cohorts of Crohn's disease and ulcerative colitis patients and healthy controls. And only one cohort, the Cincinnati cohort, had Crohn's disease patients with upper GI involvement according to the Montreal classification. So we looked into the samples and we saw that only that cohort, the Cincinnati cohort, that had patients with upper GI involvement, had IBD patients had more dietary protein signal in their stools, suggesting that more proteins were actually reaching the stools rather than being absorbed, whereas the Mass General cohort had the same amount of protein signal exactly, and this again had no upper GI involvement in Crohn's disease patients. When we looked only in the Cincinnati cohort and we compared those patients, exactly those patients with upper GI involvement, which are those, we saw that these patients actually exhibited a lot more dietary signal reaching the stool compared to patients with no upper GI involvement. And this may be, it can be postulated that this is just a protein malabsorption owing to active Crohn's disease in the upper intestine, which is where proteins are being absorbed. I forgot to mention that. So I'm going to wrap up by saying that this is an integrated proteomic genomic pipeline that simultaneously assesses post-bacteria and diet. We were able to reach an unprecedented bacterial phylogenetic resolution, and the therapeutic, the translational implications are identifying therapeutic targets, identifying probiotic resistance proteins, assessing dietary compliance and digestive function, and finding new biomarkers. This work was published in Cell this year and was achieved by the help of a lot of people, which I'm very grateful to work with, and that's it. I'll be happy to take any questions, and again, thank you for the organizing committee for inviting me and for arranging for me to talk online since I wasn't able to physically attend, unfortunately.